The overall goal of the Follow-Me experiment is to record neural spiking activity from a large number of neurons. This is achieved by observing the increase in intracellular calcium concentration in a neuron soma associated with each action potential using fluorescence microscopy. In the first step, two-photon excitation and a random access scanning principle are used to record calcium fluorescence signals from 40 neuron somata with a high spatial and temporal resolution. In the second step, the precise spike timings are extracted from the fluorescence signals using a deconvolution algorithm. This technique can be used to record spiking activity from tens of neurons. The spiking data then can be used to measure mutual information, signal propagation between neuronal populations, or neural correlations.
The main advantage of this technique over existing methods like microelectrode recordings is that one can record neuronal spiking activity from a large number of neurons from a local population.
This method can help answer key questions about cortical function on the level of local networks of neurons, such as the importance of correlations, signaling of information, and changes in network dynamics with cortical plasticity.
For two-photon excitation, an infrared pulsed laser system with femtosecond pulses is used. A high laser output power is required to offset the large losses introduced by the optical components of the system. A pre-chirper system consisting of two prisms imparts a negative group velocity dispersion onto the laser pulses prior to the acousto-optical deflectors to compensate for the temporal dispersion introduced by the AODs. Two AODs with large apertures deflect the laser beam in 2 dimensions. A reflective diffraction grating with 100 grooves per millimeter